here we are. This is the slide we left off on. And we were talking about cell division, this whole unit, right? But we were focusing the last lecture and this lecture on basically this principle of genetic material in the form of DNA, chromosomes, the coiled up, tightly packed, condensed DNA structure that we find in the nucleus of our cells. And we specifically have how many chromosomes total? See if you guys remember. Oh, there was internet outage. Nice, guys. We have 23 pairs. Oh, that makes me feel good. People are listening to me. Awesome. So we have 23 pairs. Great. So 46 total. And these are our homologous pairs. We remember I was kind of emphasizing we have 2 chromosome 1s, 2 chromosome 2s, 1 from mom, 1 from dad. So we get 23 from mom, 23 from dad for a total of 46. Okay, now what I really want to emphasize or start with today that I didn't get a chance to address is what is the difference between a chromosome 1 from mom and a chromosome 1 from dad? As we can see here from our karyotype, taken from any one of our cells, except a select few, which we'll talk about. But if one of these chromosome 1s came from my mom and 1 came from dad, what is the difference? Why are they both a chromosome 1? Why are these both a chromosome 2? You know, why isn't it not like chromosome 1, 2, 3, 4, 5, 6, etc., right? Well, that's what we're going to talk about. They're homologous chromosomes. And from last time, we talked about how they are similar, but they're not identical. That's what we're going to talk about right now. What the heck are you talking about, LaRue? What does that mean? How can I have 2 chromosome 1s and they look the same? How are they not the same? But they're both chromosome 1s. Now, remember, I kind of equated the chromosomes to an encyclopedia. So they range in their information. Oh, I got my flu shot, by the way. That's the Band-Aid. Okay, so they range in information from like A to Z, right? You might find a gene for insulin, DNA for insulin on chromosome 4. You might find DNA for a structural keratin protein in hair on chromosome 20. Those are just random numbers, but you get the idea, right? Let's take a look at the difference, very subtle difference between homologous chromosomes, okay, these pairs, and the similarities. And you'll see what I mean when I say it's a very subtle, interesting difference. So I'm going to write homologous pairs I'm going to write, how are they similar? How similar? Let's use purple. I don't use purple very often. So let's write, how are they similar? these homologous pairs, these 23 pairs. They're the same size. That's for sure. That's one. And my second criterion in my notes says carry similar genetic information. But I'm going to be a little more specific. And I'm going to say carry same genes at the same locations. Carry same genes at the same locations. Okay, what does that mean? What does that mean? Let's take a look at what I mean by that. What this means is that, let's say chromosome 5 here. Let's say this is my paternal chromosome 5 that I got from the dad. And let's say it has like, 300 genes along the chromosome. 300 genes, a gene being a region of DNA that codes for a trait. And so if I got or looked at one of those genes and it's, say, the gene for a digestive enzyme, and it's located right here. This is the gene for my, let's say, lactase, digests lactose enzyme. And the gene for that's right here. I am going to find that gene on the other chromosome 5 at that same spot. They are similar in that regard. You will find the same genes on chromosome 5 from the dad as you will on the chromosome 5 from mom. Same genes. Now here's the subtle difference, okay? They carry the same genes at the same locations, but they can be a different version of that gene. I think my favorite example is hair color. The pigmentation for hair color and the gene for hair color. Now, I have brown hair, and let's say my friend Danny has blonde hair. The gene for hair color will be on the same chromosome as Danny, but do we have the same color hair? I'm sorry. Let me backtrack. I didn't phrase that correctly. Okay. Would you say that Danny and I have the same gene for hair color? Yes or no? Do we have the gene for hair color? You guys get where I'm going. Absolutely. We have the gene, but it's a different version, right? His hair color gene codes for blonde hair. Mine codes for darker brown hair. But it's the same gene. It's a hair color gene. Think of another example. Height. Say I have a gene that codes for height and how tall I will get. And then I have a friend who's much taller than me. They also have a height gene, totally, but it's a different height gene, right? It codes for a taller human being. This is what I mean by same gene, but different versions are possible. And that's the difference between a homologous pair. You're going to find the same gene at the same location on both of these chromosomes, but they can be different versions. So for that enzyme example, you could have that digestive enzyme coded for on one of the chromosomes, but then on the other one, it might be a defective version of it. It might be mutated, faulty, and non-functional. But it's still, the gene's there. It's just the wrong version. It doesn't work. It doesn't function, the protein, the enzyme. But you'll find that same gene. So I had a student come up with this really awesome analogy too that I love to use the other semester. And he said, it's kind of like if you're buying ketchup, right? And you can get Heinz ketchup or Hunt's ketchup. They're both ketchup, right? We're still talking about ketchup, still talking about hair color, but it's a different version. It's a different company, different flavor. And they taste different. I didn't even know. I can't tell, but right. And it's a different version, but you're going to find that same gene. It's just not identical. It can be though. So that's the weird part. It can be identical, but it doesn't have to be. That's the point with homologous pairs. That reminds me, I need to pick up ketchup. Get ketchup. I'm all out. I like using my own ketchup when I get like burgers and stuff. Okay. Does that make sense? Does anyone have a question about that sort of abstract concept there? It seems like you guys got it. Same gene, but different allele is what we sometimes refer to it as. And we'll talk about that more in genetics, that word. But the different version of that gene has that fancy word that we'll talk about more at a later date. Okay, now, like I said, we're not going to be talking about cell division today, but I put this slide in here because we're going to be talking about some of the terms for cell types and what these cell types are that undergo these types of cell division, mitosis, meiosis. So, and I do like to kind of put it in the back of your head of this is the main story. This is the main topic for this unit is the process of cells dividing. And there's two types. You have mitosis, and this is going to result in two brand new cells called daughter cells that are genetically identical to the parent cell, the original cell. Meiosis, on the other hand, is this different, unique form of cell division that occurs in only one cell type, and these cells will perform meiosis to generate gametes, sperm and egg cells. And the process results in 4 genetically unique cells. Now, you don't have to worry about the details at this time. We're going to really hammer out how exactly this goes, how exactly these processes occur, okay? But let's see. For now, this lecture, we're focusing on learning new vocab words, really, and some conceptual stuff. So we're going to talk about what is a somatic cell, what is a germline cell, and what's a gamete cell. And the reason is because they all relate to these processes of cell division. And you'll see that in a minute. So let's start with a somatic cell. Write out the definition for what we mean by a somatic cell. Okay. Somatic cell. Soma, the Greek root soma actually means body, the body of something. So it's a body cell. It's actually most of our cells, like 99% are somatic cells. So examples include our skin cells, liver cells, intestinal cells, etc., brain cells, you name it. These are our somatic cells, body cells. Like I said, we're learning new words these 2 lectures. And a very important term is diploid. Somatic cells are all diploid, and I'll define that. So somatic cells all are diploid. Did I define diploid already? I feel like I might have talked about it. I did on the last lecture, maybe? Did I? Briefly, okay. Okay, cool. I'll talk about it again though. I'm glad I actually mentioned it though. Diploid refers to the beginning of this lecture recorded. Yeah, it should be. Briefly. So I think, okay, I'll just redefine it. No worries. Essentially, diploid refers to a cell or an organism that has 2 sets of chromosomes. 2 sets, just like we have a set of 23 from mom and 23 from dad. We have 2 total sets of chromosomes, right? So I'll write for humans, total of 46 chromosomes. Oops. Slash 23 pairs, right? This is 2 sets. And I'll go a little further here by, so we actually use this terminology here to define a diploid cell or organism with diploid cells. We say 2N. And so if you see 2N, you know we're talking diploid. And then what we'll do is say 2N equals, and then we'll write the total chromosome number. So for humans, that's 46, right? So we call this the diploid number. This is the diploid number. And the N refers to the number of chromosomes in a single set. So N would be 23, right? And so we have 2 sets of 23 to get 46. So this is the total chromosomes. So the other key thing here is that all of our somatic cells are diploid cells. They have 2 sets of chromosomes, total 46. So any one of these cells is going to have 46 chromosomes in it. Okay. Our next cell type is a germline cell. And a germline cell, what I want you to know about germline cells is that they're also diploid. That's important. But they're special stem cells. So they're diploid stem cells that reside or they're located for males, the testes. So in testes, this is for males. And then females, the ovaries. Or ovaries for females. I ran out of room, but just in your notes, just right below germline, I'm just going to erase up here and keep writing. So our germline cells are the ones that are going to do meiosis. Meiosis. That's why they're important. So I'm going to write, it divides, germline cell divides by meiosis to produce gamete cells. That's our next cell type we're going to talk about. I'll use green. So germline cells do meiosis and produce gamete cells or gametes. And I mentioned it, but our gamete cells, it's just the fancy word for eggs or sperm. Depending if you're male or female, you're producing eggs or sperm cells. And the important, interesting thing about eggs and sperm cells, they're not diploid, like the germline cells and like somatic cells. They're actually haploid, another new word. So they are haploid. So let me write haploid. And it kind of sounds like half, right? Diploid versus haploid. Haploid is, it means you have one set of chromosomes, not two. So you only have one chromosome 1, one chromosome 2, one chromosome 3, etc. So how many total chromosomes would a gamete have? 23, exactly. And so we call that the haploid number, right? It's not 2n, it's just n equals 23. This is the haploid number. And like I said, when we talk about meiosis, we're going to be starting with a diploid stem cell with two sets of chromosomes and produce gamete cells with less. So it's a pretty incredibly, interesting process of cell division. Okay. So the next thing I want to talk about is something that occurs prior to cell division. and it's important. And we're going to learn some more new words. It occurs before cell division, an incredibly important process where if you're going to divide your cell into two, you're going to have to copy all of your DNA so that both of your new cells are genetically identical, or they have the same amount of DNA. So we're going to talk about how that happens. So we call this DNA replication, when we copy all of our DNA and duplicate it into an identical copy. So we duplicate the chromosomes, right, with the DNA in it, or in them, or replicate our DNA. So let's talk about what that is. And first, while I'm thinking about it and looking at my notes just now, I want you to take a quick look at these chromosomes, okay? And notice how they're single-stranded. We call these single-stranded chromosomes, okay? Just keep in the back of your mind while we do this process, these single-stranded chromosomes, these 46 chromosomes, are not duplicated. That's what I was going to say. They're not replicated. This is before these chromosomes have copied the DNA contained within them into an identical strand or copy. So this chromosome 1 is different than this chromosome 1, right? They're homologous, similar, but different, as we learned. Just keep that in your mind while we talk about this, okay? Those are unduplicated. And that's where we're going to start. A homologous pair that has not duplicated yet. And so I'm going to use a red to denote the maternal homolog, and a blue for the paternal homolog. Like that. So this is paternal. This is maternal. This is a homologous pair. And I'll also take this opportunity to teach you another word, centromere. Centromere is just the word for the center region of the chromosome. So these are centromeres. And then I ran out of room here, but this is two separate chromosomes, right? We're going to be talking about another subtle distinction here in a moment. But this is two different chromosomes. You remember a chromosome, forget if I said this, but a chromosome refers to a unique subset of DNA, unique. So this is two unique subsets. These are different from one another. It's maternal and paternal. Same chromosome number, but as we know, they're different. Okay, now let's replicate. Let's do DNA replication. Let's copy all the DNA in these two chromosomes. This is what's going to happen. And forgive my awful artistry skills, but I will do my best here. You know, it's actually better to draw them like that. Kind of separate, not like one big blob. Okay. They form these two X shapes. We call these double-stranded chromosomes, while those were single-stranded. Okay. Now, based on what I said about what a chromosome means, okay, as a unique subset of information, if I duplicated, copied exact identical, the entire length of this maternal chromosome and attached it at that center region here to the, you know, original, I have this exact identical copy, right? And then I did the same for my paternal chromosome. How many chromosomes do I have now? It's a bit of a trick question. I don't have 4. I don't have 4. Remember, it's a unique subset. I still have 2. Yeah, that's where it gets tricky. I still have 2 chromosomes. I have the same amount of information as what I started with and ended with. No new unique genetic information. That's the definition of a chromosome. What I have are now sister chromatids. So these are 2 identical, what we call sister chromatids, attached to this this one double-stranded chromosome. And then we also have 2 sister chromatids here. Sister chromatids are identical. So I'll add identical, right? Identical duplicates. Well, like I said, I have not added or taken away any DNA that's different. So I can't say I took away a chromosome or added chromosomes. We just went from single-stranded chromosomes to double-stranded. So I'm going to write where can I fit it? I'll do it here. End with 2 double-stranded chromosomes and I'll add with 2 sister chromatids each. Running out of room there, but this is what I'll move this up to. So after we duplicate our DNA into identical strands, sister chromatids, we end up with 2 double-stranded chromosomes, and each chromosome here has 2 sister chromatids. So we have a total of 4 sister chromatids, right? So it's a bit of a trick question, a little weird, because we started with 2 chromosomes and we still end with 2 chromosomes. That's why I talk about this, because it can get tricky, right? Okay. Now let's practice this concept. Let's do a practice in class assignment here. In groups of 2 or 3. Well, not really. Okay, so if I tell you that the diploid number for dogs is 78, how many total chromosomes would be found in a somatic dog cell? Don't overthink it. It's the total number of chromosomes. So how many total chromosomes in a somatic dog cell? Remember, somatic dog cells are diploid. They're diploid. They have 2 sets. So let me first tell you that human beings have a diploid number of 46, and we have a total of 46 chromosomes. Correct, 78. Yep. So somatic dog cells are diploid. This is the total chromosome number. All this 2n means is that there's 2 sets for a total of 78. Total chromosomes, okay? Now, how many would be found in the gamete cell? 1 set. 1 set of chromosomes. Yeah, that's 39. Yeah. So exactly. So 1 set is 39. If you have 2 sets, that's 78 in the dog's somatic cell, or human somatic cell would be the 46, right? And then in our gamete cells, we have 23. We have half, and the dog has 39. It's 1 set, totally. Okay, how many sister chromatids would be present in the somatic dog cell after replication? So we know the somatic dog cell has 78 chromosomes, right? After replication, we just learned that it still has 78 chromosomes. How many sister chromatids? That's right. 156. Yeah. Yeah, they just all the 78 chromosomes do this. So the dog had 78 of these, 2 sets total of 39, 39 of these, 39 of these. And they all did that. So you just multiply by 2. They all were single-stranded. They all became double-stranded chromosomes. So each one has 2 sister chromatids. If you have 78 chromosomes, times it by 2, and you get the total amount of chromatids. Now you can see why I spent a whole lecture, 2 lectures really, on these concepts. It's all very interesting, complex stuff. Okay. Questions. So does each, Sophia, that's correct. Only after replication, when they become double-stranded, right? Each chromosome has 2 sister chromatids that are identical duplicates after DNA replication. But before, there are no sister chromatids, right? Attached to each other like this. Yeah, so this is 4 total, exactly. Yeah. If you have a homologous pair that does this, you'll end up with 4 total sister chromatids, yeah. What does the centromere do? Nothing. It's just a location, and it's essentially it's where the sister chromatids attach to one another. It's where in mitosis and cell division, there's going to be these spindle fibers that move chromosomes around, and they attach at the centromere. I just want you to know that it's this location, that's in the center. It's the center region of chromosomes. So that, you know, if you're reading the textbook, it's going to say like, oh, it attaches at the centromere, or sister chromatids are attached at the centromere. Yeah, they came up with a name for the center. Okay. So I believe let's see. Okay, so the rest of the lecture is a bit different. We're going to be talking about conceptual stuff and reasons as to why cells divide by mitosis, not meiosis. We'll get there. But we're going to talk about why cells divide by mitosis. And then we're going to talk about the cell cycle and what is cells doing in its life before and after mitosis and meiosis, depending on if you're the germline cell. But for the rest of this lecture, we're going to look at why a cell would do mitosis and divide, and why a cell does certain things in its life. We call it the cell cycle. So let's begin by talking about why cells divide by mitosis. Okay, and actually won't be writing on the board for these few slides. Probably the most, I wouldn't say important, but one of the most striking reasons why an organism would divide by mitosis is growth and development. And importantly, all multicellular life develops from a single cell. You, me, everyone you know, started as one cell that divided mitotically by mitosis over and over and over and over and over again, until trillions of cells later, you are here and we can see you. And this is all done by mitotic division. So that's a pretty important one. Next reason, repair and regeneration. So constantly, humans are having cells divide by mitosis, but only certain areas. Only certain areas, really three prominent areas where cells are actively dividing. And that's skin cells. I think it's about a week maybe, but skin cells are constantly shedding off and replacing what we call the epithelial layer of skin cells every week. So they're dividing pretty rapidly, constantly. And we're shedding these microscopic skin cells all the time. So a lot of mitosis there. The intestinal cells, maybe that was the 4 to 5 days statistic. So the lining of your intestines is constantly replaced every 4 to 5 days with cells that are dividing and replacing older cells in your intestines. So it's constant. That's a constant process as well. And then the last is blood cells. So cells in your bone marrow, actually, that's where you get your red blood cells. It's where you get your white blood cells. And those are constantly dividing because red blood cells, I always forget number stuff. 90 days they last before they degrade and die. But red blood cells constantly need to be regenerated, as well as white blood cells. So in the bone marrow, that's where your blood cells come from. You're going to find mitosis happening to regenerate your cells. Now, some animals can regenerate limbs. That's pretty amazing. So the starfish is a good one. So if the starfish loses an arm, tragically, it will divide by mitosis to produce an entirely new starfish. So we, of course, don't have this ability, but a lot of multicellular life can do this. Do I have the, oh, it's on the next one, never mind. But yeah, so this is a regenerative process. Oops, sorry. And also repair, right? So I didn't mention repair. So who's had a cut before, taken a bad fall? What happens is this process of re-epithelialization. And you're going to get new skin cells that are going to divide by mitosis and make more and more to seal up your wound. Of course, if it's too deep, can't get enough cells, you get collagen, you get a scar. But wound repair, of course, is a very important process where mitosis is involved. So repair and regeneration. Okay, and the last reason we would have cells that divide by mitosis. Oh, also another interesting side note, just really quick. Oh, my knees. So pretty much, I had a lecturer in grad school say, quote, oh, sorry, can you hear me now? I had a lecturer in grad school tell me, quote, you don't want your cells dividing. You don't want them actively doing mitosis. And if you do, it forms a tumor, it can be cancer. And so really, besides skin, bone marrow, and intestinal cells, nothing is dividing. Your brain cells, your muscle cells, pancreas, liver, nothing is doing mitosis. It's in this quiescent, we call it, state of just being chill. So it's just interesting to know that, I think. And when they are actively dividing, it's not good. Okay. The last third reason we would have cells divide by mitosis, of course, we don't do this, is asexual reproduction. So this is when a single-celled organism or a unicellular organism divides by mitosis or binary fission. It's a fancy process for a mitosis event where cells are dividing, producing two identical cells and producing clones, right? These are clones. So here, these are amoebas, or one amoeba becoming two amoebas, right? So that's a unicellular organism dividing by mitosis to produce another unicellular organism, single-celled. And then, of course, there are actually some multicellular organisms that can reproduce asexually. So strawberry plants are an example. And they actually shoot out these runner stems, as seen here, to produce new strawberry plants, but this is all through mitosis, meaning they're going to be genetically identical to the parent organism. And when we talk about mitosis in depth next lecture, we'll see why. We'll see why the process of mitosis actually creates identical offspring. Okay, I think succulent plants can do that as well, or maybe it's more of a regenerative thing. I think you could, if you cut off a succulent leaf, you can stick it in the ground and it'll grow a new one. Pretty cool. Okay. So let's talk about the cell cycle and what a cell's doing for its life besides dividing. Actually, yeah, including dividing. That'll be part of it. Let me get my eraser. Wow, that's sharp. Okay. So I'm setting up a reminder. Okay, cool. Cell cycle. Okay, for the cell cycle, let's do blue and let's do purple and red. Okay. Just going to do that. Leave a space here and do it in a different color. Do maybe an arrow. So the cycle goes like this. And there's two main phases of the cell cycle. The first consists of 3 stages or phases within it. So in purple here, this is called interphase of the cell cycle. This is where the cell spends 90%, as you can see, of its life in interphase. So 90% of the cell's life is in interphase. And then 10% of the cell's life is in the second phase here, which is cell division. And for this example, let's do mitosis, because that's the majority of cells, right? So mitosis, do here. Sometimes called MM phase for mitosis or maturation or whatever. So this is 10% of the cell's life. Okay. So because we're going to be talking in detail about mitosis next class, I want to focus on interphase for now. And the 3 phases within interphase that are part of interphase, or stages. And the first one's called G1. The second one is called S phase. So I'll put an S up here. So it goes G1, S, and then G2 before mitosis. So we're going to talk about each one. G1 stands for first growth phase. Growth phase. And what the cell is doing in G1 is it's going to grow and perform normal cell functions. So cell grows and performs normal cell functions. Okay. When the cell enters S phase stands for synthesis, specifically DNA synthesis. DNA synthesis. This is when DNA replication occurs. So DNA replication occurs. Okay, so I'm going to show you something interesting. Now, if you remember from last lecture, on the very first slide, we looked at chromatin in its uncondensed form, okay? And I told you to kind of keep in the back of your mind that chromatin in its uncondensed form, that spaghetti-like form, it looks like that in interphase. Okay, so this entire interphase, the cell has its DNA in chromatin form. Chromosomes are not a thing yet at all. But importantly, when we duplicate, as we saw, that was, it was showing the condensed version, right? But that's not what happens in reality. In reality, the cell, I'll just have a, let's do blue. It has, okay, here it has the 46, here's a nucleus, not drawn to scale, but 46 total chromosomes, but they're in uncondensed DNA form, like that. After S phase, the cell, I'll draw it here. It just looks like a little more dense, thicker bowl of spaghetti, because all the DNA copied itself and formed an identical DNA strand to duplicate all of the DNA, but it's uncondensed. Now, I tell you this because when we enter mitosis, the first thing that happens is the DNA condenses into chromosomes. Prophase. I told you to keep that in the back of your mind too, which we're going to focus on next class. This prophase of mitosis, the very first phase of mitosis, the chromatin condenses, but if it condenses after duplication, which it will, because after S comes G2 and then mitosis, after duplication, we have those chromatids now. We have twice the amount of DNA. So you're going to immediately see those X-shaped chromosomes with sister chromatids. And this is why, because we have duplicated all of our DNA. I just like to emphasize that in reality, in interphase, it's uncondensed. I think a lot of professors say like you have chromatids here, and that's not true. It's not condensed into chromosomes. It's still uncondensed chromatin state. Okay, so that's S phase. DNA is being copied like this. And then G2 is our second growth phase. And this is really a phase devoted to preparation for cell division. So cell gets a little bigger, it's a growth phase. Cell gets larger. And we'll write the cell prepares for division by actually duplicating organelles is one of the things it does. We'll just write that. So all the organelles get duplicated as well. Okay. Mitosis, this is cell division. We're going to have one cell become 2. Move my phone here. Gonna have 2 cells, and actually mitosis is often coupled with the word cytokinesis. So I'll write for the third sort of event, cytokinesis. This is just referring to the splitting of the cell. The division of the cytoplasm is cytokinesis. This is the division of cells' cytoplasm. We'll be talking about it again next time. Just one more thing I want to talk about before the end of lecture today. Some water. Any questions? Okay. Done writing? Oh, actually, I'm going to keep this up. Totally forgot, but that's okay. I just want to keep up the cell cycle part. Because there's a few cell cycle checkpoints that we're going to talk about. Okay. Now I think it's upside down in my slide, but that's all right. It's the same things, but okay. So the cell cycle has to be regulated. There needs to be some control mechanisms in place, checkpoints, and there's 3 in particular. Here's our cell cycle. Like I said, mine's drawn upside down. I have M phase on the below, but it's the same order, G1, S, G2, mitosis. And there's a couple checkpoints. And the reason this is important is, like I mentioned with the cancer thing and what my lecturer taught me, cells you don't want them dividing uncontrollably. It forms a tumor. And in fact, in cancer, these control mechanisms that halt the cell cycle are out of whack. They're not functioning properly. They're not preventing cell division when they should. So there's a couple important ones. There's the G1 checkpoint. So let's do it in blue. And we'll do it in blue. So there's a G1 checkpoint. This checkpoint actually makes sure, or rather the cell checks for proper nutrient availability. No nutrients around, it's not going to grow. There's a checkpoint at the end of G2. So right before, oh, wait, sorry. End of G2 is down here, right? This is our G2 checkpoint. Come on, all right. What are you doing? So the main purpose of G2, because G2 follows S phase, where all the DNA is copied, we want to make sure that the DNA was copied correctly in G2. So in G2 checkpoint, the cell checks for accurate DNA replication. Okay. If there's a small error in DNA replication, the wrong nucleotides were synthesized, and it's not exact, the cell's going to repair it. There are repair mechanisms in place, and it'll repair. But if the extent of the damage or the error is so large, the cell will actually trigger a mechanism of self-destruction called apoptosis. Apoptosis, which is cell death. We don't want cancer forming. I ran out of room, but this says apoptosis, cell death in parentheses. Apoptosis is just the fancy word for how would I say? It's going to pop itself. Kind of, yeah. Yeah, it's a very organized and can't think of the word. It's the cell's decision to do this. It's not, some sort of inflammation or pathogen causing the death, like a bacteria thing or a wound or anything like that. It's the cell's own decision to undergo this mechanism of self-destruction and recycling of the cell's components because it had the extent of damage was too great. Okay, and the last checkpoint, the last 2 minutes here, is actually during mitosis. It happens at metaphase. So this is a metaphase checkpoint. Of course, metaphase we're going to talk about next time because it's during mitosis. And you won't understand what this means yet until we talk about mitosis, but you'll know next time when you look back at it. But essentially during this metaphase checkpoint, the cell checks that spindle fibers, definitely I'm running out of rooms. Erase here. Spindle fibers are correctly or properly attached to sister chromatids. Correctly attached to sister chromatids. All right, that's it for today. So any questions? Any questions? I'll read this again because it's kind of small, but the metaphase checkpoint checks that the spindle fibers are correctly attached to sister chromatids, and that'll make sense. You'll know what I mean by that next time. questions. Okay. Oh, these were. Yeah. Well, so Glorian, metaphase is actually a phase for in mitosis. That specific cell division. All right, so let's take a 10-minute break. Come back at 4:26-ish and talk about this week's lab. Cool? Sound good? Coolio.